Glaxosmithkline, enabling people to do more, feel better, and live longer.
Thank you for calling Medical Information. My name's Donna. I'm a pharmacist. May I have your name, please?
I'm calling regarding the Pediatrix vaccination and some of the inserts. Can I go through that with you?
And you wanted to talk about the Pediatrix prescribing information?
No, particularly the inserts. Just I'm a little bit confused. Not confused, but just I want some of the things explained to me.
Okay. Mhm.
So I'm on the Glaxosmithkline website, and I have the inserts in front of me here. So this is the diphtheria toxin is produced by growing Corynebacterium diphorate in phantom medium containing a bovine extract. What's that?
Well, okay. So bovine, of course, is beef. So that's from, some of our vaccines are from animal origins. That could be porcine, pork, bovine, beef. Some people have allergies, and so we continue. We have ingredient. We have the package insert contains ingredient quantities in case there are allergies or whatnot. So this is from the beef extract, and it's just saying that, the ingredients, of course, to make the vaccines, the diphtheria toxin, because you have to, it's a combination of the different antigens and the toxins that is going to go into the works to produce these vaccines,
Okay.
To make people resistant, of course, to getting the diseases. So that this is extracted from the diphtheria toxin, and they produce it by growing this organism, Corynebacterium diphtheriae, and then the medium, of course, like I picture in my head like a petri dish, if you will, a phantom medium, and it contains this beef extract, and that's how they grow it using, a beef extract from an animal source.
Okay, so would the beef extract be in the vaccination?
Well, it's this one in particular. Let me just see. This one in particular, let me just see, because usually this is just, for instance, in this one, it's just used in the processing. Sometimes, I'm just talking in general, sometimes the bovine would be in both the process of making it and the manufacturing and in the other parts of it, but a lot of times it's just in this growing process. So now with this one, for instance, and this is a couple of parts, obviously. It's diphtheria, tetanus, and pertussis. Mm-hmm. Whereas like Energix B is just hepatitis. We have some, of course, 3-part vaccines and some 1-part vaccines, that kind of thing. Mm-hmm. This one particularly looks like it's just, I'm just looking to see. Looks like it's just in that process point. It doesn't look like in the final product.
What is you don't know if the beef extract is in the final product?
Well, I'm going through to look at it because we deal with about 20 different vaccines, and we have different letters and whatnot. Right. So I want to make sure that, and I deal with all pharma products, so I handle about 90 products. So I want to make sure that I give you the correct answer on this. I want to look and see, make sure. Let me just check in one other database.
Yeah, it's just I wonder if that's in the insert, or is it removed somehow?
Well, the insert does, every package insert for the product does contain in section 11 all of the ingredients of that vaccine.
Right.
So it does contain the ingredients. Now, as a healthcare professional, you can always call us, or we do have a click to chat, which is an online sort of database where you can always email us and request questions, and you have a single one of us, an agent, a nurse, or a pharmacist working with you individually, that we will email you back the answer to your question, or and then also can get it to you as me talking to you here. I can email or fax you data as well, according to, it's basically get the information to you in writing. Let me just see raw materials. I'm just looking at this for pediatrics. It's a different database that I'm looking at.
Right.
But basically, the prescribing information has a lot of information. But if you're like zeroing in on, like for instance, I'll have a pharmacist call me and they have a patient standing in front of them that has, let's say, a bovine allergy. So they're ready to give, let's say, a flu shot, and they said, listen, I looked at the package insert, but I want to make completely sure that there's no bovine. So I'll go and scan it, and I'll look at other documents and kind of make sure. You know what I mean? Like, just to make sure. Okay, so for instance, with your question, for animal origin pediatrics, I'm looking at animal origin, including a non-exhaustive list of bovine, equine, and porcine-derived materials are used as raw materials in the manufacturing process, and that's prior to the formulation step of pediatrics. And the ingredients of animal origin are not used as excipients in the formulation of pediatrics, and the presence of ingredients of animal origin in the final product has not been verified analytically. So with all of our products, we can give a little summary of, what, like we've talked about, you and I, the manufacturing process and excipients, but most of the products are not verified analytically in the final product. So does this make any sense to you?
Okay. So there is the bovine extract is in the vaccination then?
It's used in the raw material.
Okay. All right. Okay.
In the manufacture. So that's the only that's the only place that the bovine's used. It's prior to the formulation step for pediatrics. Yes.
Okay. Well, that's good to know. All right. I'm just
and I could send you out this letter.
Right. Okay. So just let's continue through this. The bovine materials used in these extracts are sourced from countries with the U.S. or within the USDA. The USDA has determined neither have nor present an undue risk for bovine spongiform entomology. The toxins are detoxified with formaldehyde, concentrated by ultrafiltration, and purified by precipitation, dialysis, and sterile filtration. Now, something jumps out at me there is formaldehyde. That's a toxic chemical, is it not? Well,
I mean, it is a preservative. Now, some in your prescribing, in your package inserts, which are on, of course, gsksource.com, I have noticed when I've gone in and looked at some, there is a mention of formaldehyde in a couple, but it's used in it, at a very scant amount and then purified. So, of course, you're not going to, be injecting patients with something that's toxic. I mean, a lot of these vaccines, of course, have been used for years, but I do see it once in a while in some of the, package inserts. So I do see that formaldehyde. So in some of the vaccine package inserts, I can't name off the top of my head. But
if you scroll down, this particular vaccine says this vaccination contains 100 msg of residual formaldehyde. So it is in the inserts that there is formaldehyde in the vaccination. And that would it's less than 100 micrograms. That's in 0.5 ml dose. So that's a very, very minute amount. If you think of 0.5 ml, half of a cc, and it's less than or equal to 100 micrograms. Oh, well, that's okay. That makes me feel.
Yeah, that's a very tiny, tiny little dose. Yeah, very, very minute amount.
Yeah, as long as there's only a little amount of toxins in it, that makes me feel a little bit better.
Yes.
Okay, so what else is going on? Let me read this here. What else is... the cellular pertussis antigens PT, FHA, and pertussin are isolated from Bartella pertussis culture, grown in modified strainer, shallot, liquid medium. Can you explain what that is?
Are you on are you in 12.1 or you're back up in 11?
Oh, sorry, no, I'm in 11. The second paragraph.
Okay. Are you in the tetanus toxin in section 11? Are you?
Yeah, section 11, third paragraph. Sorry, excuse me.
Okay. The cellular pertussis antigen. Bordetella pertussis.
Bartella pertussis culture grown in modified strainer, shallot, liquid medium.
Yeah, I mean, I would have to look up exactly what that is, but they basically, of course, they're going to grow out that organism in whatever culture that will allow it to grow out so that they can get that antigen such that it will be able to cause immunity in the vaccine for the patient, so they will become immunized against the disease. So apparently that must have been that Bordetella pertussis culture. That Stainer Schulte medium, liquid medium, must have been, I assume it's possibly a synthetic medium. It must have been the best choice of mediums, to grow that out, the most, efficacious way to grow it out so that they'd have a good, antigen, so they'd have a good vaccine. It would cause the best immunity.
You don't know where it's originated from.
I'd have to Google that Stainer Schulte medium to see what exactly it is. Either that or I could escalate you to one of our vaccine medical information scientists if you wanted to try and get some.
Yeah, I'm just trying to get I'm trying to get to the bottom of this. Could you put me maybe to one of the scientists or?
Okay.
Is that right?
Yeah. Sure. Hold on just a moment, please, and let me see who's available. Speak with you. Okay.
Right,
hold on, please.
Thank you.
Thank you.
All right.
Hi, yeah, my name's Tim. I'm one of the pharmacists that works with Donna. How are you?
I'm very well. How are you doing?
Good. I understand you had a question about some of the mediums that are used to manufacture our vaccine. Is that Pediatrix?
Yes, sir.
Okay. How can I help you? I'm not sure if I know the actual question.
Okay. Well, so I'm on the GSK website. I'm on the Pediatrix. The inserts on section 11.
Right.
And I'm just going through like some of the inserts in this vaccination. So I'll cut to the chase. Where am I now? So I'm at the cellular pertussis antigens are isolated from Bartella pertussis culture grown in modified Stainer shallot liquid medium. Can you explain what that is, or
explain what the medium is?
Yeah. Or do you know?
I think I have something that I can offer to you as a where you can actually go to get a really nice explanation of it. It's actually an article online. To be honest with you, I'm not a biologist or microbiologist or chemist. I don't know the makeup of these mediums that we use to either grow some of the antigens, some of the active ingredients, but there's actually something that was published in the Journal of Clinical Microbiology.
Right.
And the lead author is Ron Koenig. I can actually send to you in an email or a fax what the actual citation is, if you would like.
Yeah, maybe at the end of the phone call, I'll give you my email address, and you can send me whatever information you want.
Okay, sure.
So maybe I'll go on a little bit if you're not so familiar with the cells. There's something in particular here I want to talk about in the inserts. I'm reading here from this document. The inactivated poliovirus component is an enhanced potency component. Each of the 3 strains of poliovirus is individually grown in vero cells, a continuous line of monkey kidney cells cultivated on microcarriers. Calf serum, like calf, like a cow serum, and lactoboomine hydrosylate are used during vero cell culture and or other virus culture.
Okay.
So what exactly, or do you know anything about vero cells, or can you explain that process of using vero cells?
Well, there's different cell lines that propagate and grow very well, different bacteria, different viruses, and we just know that this kidney cell from monkeys actually work very well for the polio, the 3 different polio strains that are in Pediatrix. It's a cell line that is a continuous cell line, meaning it's a one-time harvesting of the active ingredient of cells, the kidney cells from monkeys. So we did it once.
Right, okay.
And so we have a large bath of this medium, and it is continuously growing. And so when we need to grow the different poliovirus cells for a batch of vaccine, we'll take a portion of this master batch of the vero cell line.
Right.
And after we take a certain amount of probably gallons of volume of that vero cell liquid, since that master cell line is continuously growing, it will replenish itself. So there's never a need to start from scratch,
right,
if you will, or sacrifice more animals or anything like that to continuously grow the cell line for the vaccine components. It's a one-time. We did this in history decades ago, and that same cell line has continued since that day. So I know about the cell line. I know how we deal with the cell line. I don't know the chemical or biology makeup of the cell line, other than it is from kidney cells from monkeys. That's part of the cell. That's the organic component of the medium that grows the antigens, the polio viruses that we use. There's other stabilizers and pH adjusters and things like that are also included in that cell line. Those I'm not exactly sure what they are, but I know we do have stabilizers. I do know we adjust the pH, and we do have other things in that cell line other than just the kidney cells.
Okay. Well, the fact that that these cells have been perpetuated for many years, will there be a concern about the health risks of these cells?
About the health of the cells?
Yeah.
The viability of the cells?
Mm-hmm.
Well, yes, absolutely. Each part of the step of the vaccine production is, in the general sense, very fragile, I guess is the way to put it. We pay attention to every step of the process, because for a large part of the process of growing vaccines, producing vaccines, you have living cells. You have living viruses, living bacteria. So because you have a living, growing entity that you need to have a certain virology, a certain chemical makeup, a certain biological makeup, and a certain health, if you want to say that, of the viruses, you have to have a certain quality yield, and each part of the process to have that yield is, absolutely important. So yes, if the quality of that cell line were ever to be compromised, we would have an issue. Yes.
Okay. So,
my understanding to this point, we haven't had an issue, but we do monitor it. Absolutely.
I'm sure you do. I was in contact with one of the lead scientists on the Immunization Services Division at the CDC, and he actually emailed me. I emailed him about the MR5 cells, the WI38 cells, and the vero cells, and he said that because by their nature having been, I'm quoting him here now, because by their nature having been perpetuated for many years, they have potential to cause cancers. However, no such outcome has been documented from persons vaccinated with vaccine derived from these cells. He kind of admitted there that there is a chance of, there is a potential to cause cancers, and that would be my concern.
That is something that I know that our biologists that are responsible for the cell line that among including other issues that we constantly monitor for are absolutely taken into account.
Yeah, no, I'm sure there are. Yeah.
But now, if the next question is how do they do that? That I don't know either.
Yeah. Yeah. How do we know that? For example, or a question I have. So these Vero cells, is these monkey kidney cells or any traces of the DNA in the vaccination in the final product, or are these Vero cells removed entirely, or is that possible?
There are steps to what we call washing. Certain steps along the line of growing these vaccines, including Pediarix. There are ingredients that are introduced into the suspension that we are actually producing as we produce each step, and the impurities or the ingredients that we do not want in the final vaccine, the Vero cells would be one of them. We have washing steps where we eliminate or try to eliminate these impurities. So any kind of proteins left over from the Vero cell line, any kind of DNA, if you would, DNA, proteins, anything that's from the monkey itself, from that cell line, we try to wash out and eliminate it from the vaccine.
I'm sure you try, yeah, but that's not very conclusive. I'm sure so even if there's nanoparticulates of this monkey kidney cell in the DNA, that's very concerning for me, even if it's tiny amount. From my research too, they say it's impossible to get all traces of the DNA out of the vaccination. So I don't know how you would be able to study the effects of a kidney DNA and the links to cancers. I don't know how you would go about proving that, or I don't know how you would go about that.
There are release testing for each batch. So before the vaccine's on the market, we have standards and limits of certain ingredients that are in our vaccines, whether it's this ingredient or anything else that we use and including what are the limits of the active ingredients, the inactive ingredients, the stuff that we try to wash out, the impurities. We do have final batch testing.
Right.
And each testing of each lot of vaccine, each lot of the vaccine has to pass these release tests. Right. They have to either be a certain potency for the active ingredients. You have to have a certain amount. You can't have too much. And then anything that is an impurity that we try to remove, this would be one of them. We have a test, an assay, looking for the presence of it, and whether it is undetectable by an assay. If it's undetectable, you're right. It just means that it is to a level that we can't detect it on an assay, but that doesn't prove that it's not there. That just proves that the sensitivity of that assay test has not shown that there is a measurable amount.
Yeah.
So a measurable amount for some things could be parts per thousands, could be parts per million. So they would be in what we call trace amounts. Wouldn't be an amount or a measurable amount, but they are whether they are totally removed by the process or removed to a concentration that we term trace amounts. That would be on the release testing, the batch testing. And if it fails, then we don't sell that product. That product would be quarantined and probably destroyed and never be given to the general public. Now, the amount of if a batch passes all those tests, including the trace amounts or undetectable amounts of the Vero cell line, that's all FDA approved and part of the process of identifying the safety of the vaccine, long-term, short-term safety of the vaccine, and with FDA approval, then we have at least the assurance of that process, which is a stringent process, that the product is relatively safe and effective for use in the U.S. market. So that's the standard.
But the FDA has approved a lot of, like, for example, pharmaceutical drugs that a year later they prove that it's been causing all sorts of neurological disorders in people, and things have been taken off the market. So it's not like everything the FDA approves is 100% safe. I'm not saying they do it on purpose, but sometimes people make mistakes and things get approved that shouldn't be approved. So that will be my concern, that we're injecting monkey DNA, even traces of it, into our children, and that could cause some effects.
Well, the vaccine's been on the market since 2002.
Mm-hmm.
So we have about 14 years of history of the use of the vaccine. I don't know. I don't know off the top of my head how many millions of children have received the vaccine, but
has there been long-term research or studies? Like, have you been studying this for 14 years on children, or?
Sure. We're required to do, of course, before the vaccine goes to market. Any drug or vaccine goes to market, we're required to do safety testing, but we are also required, after the vaccine or drug is on the market, we're talking just about pediatrics, since 2002, we're required by the FDA to not only monitor the safety of the vaccine doses and all the safety reports, but we're also required to send those reports to the FDA. So It's not just something that we have here in Glaxosmithkline and nobody can see the reports. We actually are required to submit those reports on a periodic basis so the FDA can actually review those reports as well. So yes, we have ongoing safety monitoring of all of our products from this company.
I'm sure you do.
The entire time that they're on the market. So we have done that with this vaccine specifically since 2002.
Right. Okay. So I want to continue here with the inserts in this vaccination. Calf serum and lactobumin hydrostate are used during viral cell culture and/or virus culture. Calf serum, that's cow blood, isn't it?
Yeah, it is. Yes.
Right, and would there be any traces of that in the vaccination?
That would probably be the same issue as the monkey kidney cells.
The
whole conversation we had about that.
So we can assume there's small traces of that as well.
I wouldn't say that we can assume that. I would say that's a possibility.
It's a possibility. Okay, right. Okay, then diphtheria and tetanus toxoids and pertussis antigens are individually absorbed onto aluminum hydroxide. The hepatitis B component is absorbed onto aluminum phosphate. So there's aluminum in there as well.
There is aluminum in the vaccine, yes.
Right.
The aluminum is there to actually enhance the response of the immune system, the body's immune system, to the vaccine.
Right. Okay.
Aluminum is probably in the majority, if not too close to all of the vaccines that are on the U.S. market, and aluminum is actually in most vaccines worldwide.
I know. I see that.
Yeah, that's a very common ingredient that is included in vaccines.
But don't you know that aluminum is a toxic chemical?
Aluminum is?
Yeah. I could pull up probably 200 articles published in the pediatrics saying that aluminum is a toxic chemical associated with all sorts of diseases, breast cancer, neurological disorders, and I find it disturbing that some of these ingredients are put in these vaccinations. It's just, the more I read it, it sounds like I'm in a bad dream or something. It really does. Like, I don't understand why there needs to be aluminum in a vaccination, and like putting it into children at such a young age when children their immune systems aren't ready, and they're putting these toxic chemicals, like a concoction of all sorts of things, into this vaccination and inserting it into children.
Well, there's not that many articles or commentary on our website about aluminum specifically, and about all the other ingredients, because we don't have that service. But I can tell you there's a lot of resources out there that actually explain and support all the ingredients that are including aluminum. One of them is Children's Hospital Philadelphia. There's a gentleman named Dr. Paul Offit.
Oh, I'm very familiar with him.
Okay, yeah, he does a really good job at explaining not only the ingredients that are in there and why they're in there, but also the data that supports the relatively safe use in the general public of the United States, both children, adolescents, and adults. That's all I can really offer you on that regard. I mean, we look at our vaccines ongoing, not only prior to marketing, but also post-marketing, the entire life cycle of the vaccine that's on the market. And we do look for all of that. We take that very seriously. I hope you believe that we, as a company, we take the safety of our vaccines very serious. And so we have an entire department of physicians, PhDs, and they're constantly looking at these vaccines and looking for any kind of signals or any kind of adverse events that we can associate with the vaccine. We take that very serious. So.
Okay, so you mentioned Dr. Paul Offit. So Dr. Paul Offit he's also the inventor of a cluster of patents from Merck's Rototech vaccine. So he's actually a co-author of the vaccination. He's also the recipient of $350,000 grant from Merck for the vaccine development. He's a consultant to Merck's Pharmaceuticals, and he previously served as a member of the Advisory Committee on the Immunization Practices. The Advisory Committee on the Immunization Practices is a 15-member panel of immunization experts selected by the Secretary of the U.S. Department of Health and Human Services. They provide advice and guidelines to the Center for Disease Control and Vaccine Preventable Diseases. He was a member from 1998 to 2003, and he was involved in CDC recommendations about giving rotavirus vaccines to babies, and he voted yes 3 times out of 4 on issues pertaining to the ACIP rotavirus use recommendation, including voting for his own vaccine to be included in the immunization schedule. So there's a complete conflict of interest here. Like, we've got a man voting for this vaccination, and he's a co-author of this vaccination. He stands to make money. To clarify, Dr. Offit voted for the first RotaShield vaccine manufactured by Wyeth monkey-human hybrid virus approved by ACIP in 1998 to be given to all babies. It's just funny that you bring up Paul Offit. There's a complete conflict of interest there. This is one example of many of conflict of interest regarding vaccinations and Merck and Glaxosmithkline.
Well, I fully appreciate the history of Dr. Offit, and that's a poor example for you. There's a lot of other respected physicians, healthcare professionals, thought leaders, experts that also support a lot of what Dr. Offit said. But if you don't want to take Dr. Offit's commentary, recommendations, his expertise, then there's a lot of others.
Well, how can I listen to a man who is a patent holder in rotavirus? And of course he's going to be biased towards this, and when he stands to make money off it, like, there's a complete conflict of interest. You know what I mean? Like,
and if I can, if you don't mind, I understand your concerns, and I think this is kind of obvious. You have a lot of criticism of vaccines, and that's very within your right to have those. But I do have to ask if there's another question that I can help you with. I was going off on a tangent there.
Each 0.5 milliliter dose contains aluminum salts as an adjuvant, 4.5 micrograms of sodium chloride. Each dose also contains 100 mcg of residual formaldehyde, 100 mcg of polysorbate 80. What's polysorbate 80?
Polysorbate 80 is what we call a wetting agent. It's used to make sure that the suspension, actually all the ingredients within the suspension kind of flow together and make sure that there's a homogeneous suspension when you do shake it, that it's equally dispersed.
Right. Okay.
Kind of like putting liquid soap between your fingers. It gets real slippery. And so it acts as a wetting agent.
It's a chemical, is it?
It's a chemical. It's a liquid chemical.
So do you see where I'm going with this here? Like this.
To be honest with you, no, I don't know.
You don't know where I'm going with this? Like for example, when parents are getting their children vaccinated and they're getting this vaccination, they think they're just getting the vaccinations for the viruses, but they don't realize there's all these other things, inserts inside the vaccinations, and it's very difficult to even get the inserts. Like if you go to a doctor, they won't even tell you what's in the inserts, and if you ask them, they get angry at you and for even questioning the doctor.
Right. There's a reason why we prefer, now it's in the public domain, but these packages, we prefer that a healthcare professional with knowledge of the ingredients actually does discuss all of these things with people that don't understand these things, like mothers and fathers, only because it can be a little not understanding everything can be a little concerning, and it's nice to have. It's probably important to have somebody with a little bit of science background in order to discuss what each of these things are and be able to discuss intelligently with the people that are going to get the vaccines what all these things are. And so that's the reason why. It's no other reason really than that. I mean, the information is a little difficult to understand, especially if you don't have a good science background or a chemistry background, things like that. So that's the reason why. There's nothing in these prescribing informations that are a secret that needs to be hidden from the general public. It's just that the documents are very technically oriented and a lot of technical information that it would confuse most people that would read it.
Well, do you think people are too stupid to understand what aluminum, formaldehyde, monkey kidney cells are? Do you think people are too dumb to understand what that is?
I would question if people have enough of a background to understand the impact or the non-impact of it. I think everybody knows what aluminum is, but I don't think an average person would necessarily understand the implications of having that aluminum in the vaccine. So that's really where I think it's nice to have a discussion with somebody that presents the prescribing information, presents that information to a mother or father. Whether they're intellectually capable of understanding it, I wouldn't be able to comment on that. But
okay.
I think yeah.
Okay. I'm going to finish off the inserts here now.
Sure.
The procedures used to manufacture the HBsAg antigen results in a product that contains less than or equal to 5% of yeast protein. So there's yeast protein in there as well?
There is yeast. The yeast is used to, as the virus cells are a good medium to produce the polio virus, the yeast cells we found is a good medium to produce the hepatitis B component of the vaccine.
Right. Okay.
That's a process that's been 25, 30 years, something like that. Don't quote me on that, but it's been more than 20 years. I know that.
But people keep telling me, we've been doing this since the 60s. That doesn't mean it's safe. Heroin and cocaine has been around for a long time, but that doesn't mean that's safe. I just don't understand that theory of thinking. Like it's been around 20 years, it must be safe.
Well, only in the context that we've had FDA approval that it's relatively safe prior to market, and then afterwards, as I mentioned before, we did continuous safety monitoring on all of our vaccines. So when I mentioned that a certain amount of time frame, that affords us the luxury of having a larger and larger pool of data to pull from, and that's what's important. So when I say, yeah, I agree with you. Necessarily, not just the time frame doesn't necessarily prove. It's just that the amount of data that we can gather within that time frame is the important part. So I didn't mean to misrepresent.
No, it's okay. So regarding the Vero cells, it's quite a controversial cell to be using, I guess. You're saying that's effective, but is there any research, or can you show me some research to show that it is safe?
I can also search for that for some data, or some published data, published reports,
right?
Maybe even a published study. I can look at that. If I find something, I'll put that in the same email that I'm going to send to you.
Right.
With the journal from that I'm going to give you in clinical microbiology, and I'll search for that topic as well.
All right, that sounds good.
Okay.
Thanks.
All right, Niall. You're very welcome. Have a great day.
See you later. Bye.